We have definitely a topic that everybody is going to be interested in. We're going to hear about small cell lung cancer research from Dr. Trudy Oliver. She is from Duke University School of Medicine and Duke Cancer Institute. And in the essence of time, we are going to hold questions until the end. Okay.
Good morning. It's nice to see so many of you. I'm going to just go ahead and give you a preview with this cartoon that the science I'm going to tell you about today is our view that small cell is having a bit of an identity crisis. For many years, if you read any review on small cell, it will say that small cell arises from a cell called a neuroendocrine cell because it is a neuroendocrine cancer. But some discoveries in our lab have recently convinced us that we've had it wrong for decades, and that small cell actually arises from a different cell type in the lung called the basal cell. So Bryony Hoggett in my lab, who's a great little graphical artist, made this cartoon of the neuroendocrine cell looking in the mirror and seeing a very hot basal cell, which will make a little bit more sense later, instead of seeing itself. And some of you met Bryony yesterday. She was a tech in our lab and is now a graduate student in the lab. And this picture was actually from a year ago, our event where we gave a lab tour at Duke. I think there were about 12 or 13 people from your group that toured our lab at that time. And you can see as of yesterday, the group basically doubled in size. So we had a really good time hosting you all in the lab. It's certainly very meaningful to my lab members to see you and be inspired by you and touched by you. And I've heard that it goes both ways, so we really appreciate you. And hopefully next year, for those of you who may be here for the first time, if you're interested in seeing the lab and touring the lab next year, we hope to keep up this tradition. Before I jump into any science, I want to say I'm really just the spokesperson for the lab. I don't do any of the work anymore. I don't even really know how. This is accomplished by many talented scientists in the lab. We have about 21 people now. The work that I'll tell you about today was largely led by Abby Ireland, who's a superstar grad student in the lab, with help from Bryony, who did the art on the first page, and undergrad Daniel, and collaborators that we have from all over the country. You may recognize some names that gave us human samples down below because they're very prominent lung cancer physicians. Also Joe Chan from Sloan Kettering, who helped us with computational work, and Andrew Elliott from Caris Life Sciences, which has one of the largest data sets of human small cell lung cancer available. Okay, so I'm gonna start out very broad, and then we're probably gonna get very sciencey, but don't worry about that. I'm gonna try to give you the take-home messages, so if the science is hard to follow, that's okay, just stick with me and I'll try to give you the punchline as we go. But in general, what our lab does is try to take patient data, so information that we have from real patient tumors, for example, the genes that are altered in human cancer, the characteristics of human cancers, and to model that in sophisticated animal models like mouse models. And we know this is very important to do. We know that we need living systems to best mimic cancer, and that cancer doesn't always behave in a dish the same way it does in real living bodies. And so we have the belief that it's very important to model this in living systems like mice. We sometimes initiate cancer in mice, and it looks and behaves a lot like the human disease, and sometimes we take real patient tumors and inject them into mice. And we ultimately look for agreement between all of these systems for the sort of core behavior of small cell. We have very sophisticated equipment where we image tumors in mice, much like what happens in the human disease, and we follow tumor progression over time as we test new drugs and therapeutic strategies. We also take advantage of other types of systems like organoid models where we grow tumors in 3 dimensions, and we study the cells in that environment. And we also pay attention to the tumor as a whole using things like single-cell genomic technologies, where we can ask what's happening in every single cancer cell to fully appreciate what's going on in cancer. And we integrate all of this information constantly and go back and forth with our observations in the human disease. Okay, so now I hope to teach you a little bit of lung cancer biology. It'll get progressively more complicated, but stay with me, and also feel free to interject if something just really doesn't make sense. So I like to teach my students in the lab that the lung is like an upside down tree. So imagine the big trachea and big bronchi that are leading down into smaller and smaller branches that eventually flower. And those are your airways, and where gas exchange happens in the bottom part of the tree, what gives you oxygen into your lungs. This is another example of the structure of the lung, where you can sort of, appreciate the trunk or the trachea and main bronchi branching into smaller and smaller branches. Along this tree, you have different specialized cell types that are there to do different jobs. Just like you may have bark and flowers, the lung structure differs as you go from the trachea down into the smaller and smaller airways. So you have different cell types there that have different identities or states, and they're doing different jobs. Now, let's focus on what this means for small cell. Well, of course, small cell arises in the lung, and it tends to arise in the very central parts of the lung. In the major bronchi, can you see my pointer? Yeah. In the major bronchi and these primary bronchi here and near the hilum. So that's important information, because small cell arising there means it's most likely arising in cell types that are located in those locations. So what cell types are located in those regions? So here I've sort of made a cartoon of the cell types found in the major airways, and I'm just going to introduce you to a few of them, and they're going to sound familiar to some aficionados in the room. So for example, there are these rare cell types called neuroendocrine cells. These make up less than 0.5% of the total lung, and they express this gene called ASCL1, and that's going to be well known to some as the fact that it's expressed in a high fraction of human small cell. Now, historically, the field has thought that small cell arises in these cells because they're the neuroendocrine cells, and small cell is neuroendocrine in nature. But we don't really think that that's true anymore, and I'll hopefully explain a little bit why. You also have these other rare cell types called the tuft cells, so named for a little tuft of hair they have on the top of their heads. These are PAL2F3 positive, so they express this gene called PAL2F3. They're also very rare, just like the neuroendocrine cell. You have other rare cell types called ionocytes. These are FOXI1 positive cells. These cells all just do different jobs. But an important cell type is this YAP1 positive basal stem cell. These cells are a lot more abundant. They make up more like 30% of this part of the airway, and they're positioned down here at the basement membrane, and they really protect your tissue from the things that are on the outside of the lung. So this is outside of your body where air passes versus the inside of your body. If things were to penetrate the epithelium and get inside your body, that's down here. So the basal cell layer is like a protective layer, and it's a stem cell layer. And I'll explain what that means, that it's a stem cell. Okay, so through studies, experiments in the lung, we know that if you damage your airway in some way, maybe that's an infection or inflammation, surgery, injury, inhaling toxins. When you get damage, the basal cell's job is to replace itself and replenish all the cell types that were damaged and lost under injury. So it both regenerates and repairs and gives rise to all these other cell types that you may lose. And that's why we call it the stem cell. It's really the major stem cell of the lung. I'll show you a picture of what this looks like in this really pretty fluorescent image. Actually, let me go back and make one more point about this. So we know in experiments that basal cells are the one cell type that can give rise to both the neuroendocrine cell, this is the pulmonary neuroendocrine cell, and the ionocytes I showed you, and the tuft cells. And this is going to become important because these flavors of cells are all present in small cell lung cancer. And the basal cell is the one and only cell that can do all of those things, make all of those cell types. Okay, so this is what the basal cell looks like in action. If you stain your lung epithelium, for here we're just showing two different markers, club cells, which are in red, and the basal cells, which are in green. Remember, they're located down at the basement membrane, and they're there to repair damage. So in this case, we've damaged the airway with a really strong toxin, and then we look at what happens at day 3, 5, and 10. And what you can see already by day 3 is that the red cells are completely gone. They've been completely killed by the injury. And instead of the green cells just being really small and down here at the bottom, now you have lots more green basal cells. They've proliferated and expanded. So they're getting ready, they're regenerating, and they're gonna repair the tissue back to normal. You can see by day 5, you're starting to get some of the red cells back. And by day 10, everything's pretty much back to normal. You have all your red club cells back, and the basal cells are settling back down at the bottom, right? So that's amazing. That's what our body does on a constant basis, and that's the basal cell's job. Okay, so what does this all have to do with small cell? Well, if you go to any small cell research meeting now, you're gonna become familiar with this very famous heat map pretty quickly. This is what all scientists are thinking about right now, because what we realize is that small cell is not a uniform disease where every patient's tumor is exactly the same, or even within your own tumor, every region is not exactly the same. There are different molecular states of small cell, and they can be categorized by their expression of these genes that I've mentioned and their identity, if you will. So, for example, a high fraction of human small cell, every little line here is a patient tumor. A high fraction of small cell, those in yellow, have expression of ASCL1 and are very neuroendocrine-like. A smaller group of small cell expresses neuro-D, and these tumors are a little more neuronal, which is similar to neuroendocrine, but a little bit different. And then we have a third group that has high expression of PAL2F3, and these are the tuft or brush cell-like, again, named for that tuft of hair on top of the head. And then we have a fourth, historically more controversial group that has been named all kinds of different things in the literature, everything from having YAP1 to being epithelial mesenchymal, inflamed, or triple negative for these other markers. All I want you to really appreciate here is that these flavors of small cell sound a lot like the normal cell types that you find in the normal lung, okay? So we have these normal cell types in the lung. Now, as I sort of alluded to already, when we find patient tumors, they're often not uniformly one flavor or another. They often have multiple flavors in the same tumor. And that's sort of illustrated here with this ASCL1 positive neuroendocrine cell and this PAL2F3 positive tuft-like cell. And why I'm showing this throughout the body is that this happens not just in the lung in small cell lung cancer, but in other neuroendocrine tumors you might hear about. So there's a neuroendocrine tumor called olfactory neuroblastoma. We find these tumors in the thymus, in the bladder, in the pancreas, and the prostate. Wherever you hear about neuroendocrine tumors in the body, they often have what we call heterogeneity, meaning within that same tumor, you've got these different flavors. So intratumoral just means within the tumor, there's heterogeneity or diversity in cell identity. And this is an example of what I mean. So if I take this human small cell sample, this was from Charlie Rudin at Sloan Kettering, and we section through this tumor and we stain for ASCL1, neuro-D, and PAL2F3 in these 3 different colors, you can appreciate that this tumor has all 3 colors. And they're often in what we call mutually exclusive cells, meaning the cell is either green or purple or red, but not all 3. It's one or the other, but it's a mix, okay? So you can think of the tumor as diverse and heterogeneous. We know that this is true not just in one sample, but if we look in 120 different human samples, and other groups have looked at over 100 tumors, and they stain for these different subtype or state markers, we see that every once in a while in a biopsy, you'll find only one marker, like in this Venn diagram here, here, and here. But often we see lots of overlap, meaning lots of human tumors have the expression of multiple different subtype markers in the same tumor. And we think this is a gross underestimate of the reality, because remember that when we get biopsies in the lab, this is a CT scan of a real female patient from Utah. There's a massive amount of tumor in this body, but we're just taking a little pinprick of a biopsy and looking under their microscope. So we're probably vastly underestimating how much heterogeneity is involved in the cancer as a whole, okay? So our lab was one of the first to show, and others have subsequently confirmed, that small cell exhibits what we call subtype plasticity, or the ability to change. So this is an example. The reason why this tumor is so multicolored, you can imagine lots of different colored M&Ms here, that's representing that this tumor has cells in different states, the ASCL1, NeuroD, PAL2F3 states, et cetera. Our lab was the first to show in model systems, and then we verified this through human samples, that cells can change from one state to another, okay? Others have used different approaches to come to the same conclusion. They've fluorescently labeled these different states and watched them underneath a microscope change from one state to another. Now, this is obviously very scary, because these cells in different states have different therapeutic vulnerabilities. They're not all the same identity, so they don't respond to drugs the same way. And now we know that they can change to different states, and that can be used to avoid sensitivity to drugs. So it's a big problem and challenge, and something that we want to ultimately combat. Okay, now it's going to get really a little more heavily sciencey, but stay with me. I'm going to tell you the bottom line, but I think some of the aficionados may appreciate this. So we noticed in the lab that when we were making model systems and getting samples from patients, that we had a lot of models of ASCL1 positive small cell, and we had even been able to make NeuroD positive small cell in the lab, and that was great because it gave us systems to study and understand these subtypes. But what we were really missing in the lab were we did not have very many PAL2F3 tuft-like cancer models. And this is a significant fraction of tumors, and it actually correlates with some of the worst outcomes in small cell. So in response to chemotherapy and chemoimmunotherapy, patients that have tumors with a lot of PAL2F3 tend to do the worst. And we had failed so far to really model that in our model systems. And so we were very curious why we were missing that subtype. So we wanted to take a step back and ask, what are we doing wrong with our model systems? Why don't we have representation of this state? So to do that, we took advantage of published data where it was known in either PAL2F3 positive or negative human samples, what are the genes that are altered? What are the most common mutations in the tumors that are PAL2F3 high versus low? And importantly, at the top of the list, we noted that RB and P53, these are key genes that are tumor suppressors that stop cancer from happening. These are often mutated, but they're mutated in all of small cell, irregardless of what type you have. So that was satisfying to us because we always alter RB and P53 in our model systems. We also noticed that two other genes were more highly altered in the PAL2F3 positive state, that being a gene called PTEN, which tended to be deleted in these patients' tumors, and a gene called MYC, which is an oncogene, and it tend to be amplified in these patient tumors, meaning you have more of it, more MYC, and less PTEN. And this was curious to us because we had already altered these genes in our model system, but we didn't get the PAL2F3 positive state. So that didn't make a lot of sense to us, and we had to think harder about it. Now, we verified this data from this study in a totally separate study, so this is just another way of looking at the same data. So a different Chinese group had taken a bunch of human tumors, and we grouped them as either PAL2F3 high or low, you can see in this graph. And then we looked at some of these genes that were predicted over here, for example, MYC and PTEN, and you can see very satisfyingly that the data was consistent. So if the sample was PAL2F3 high, it was more likely to have MYC amplification. If the sample was PAL2F3 high, it was more likely to have a PTEN deletion. So this is just saying across studies, the human data was very consistent that these seem to be genetic events that are associated with this state. Okay, so why aren't we getting this state in our model systems? Well, we went back to lung development and we said, well, in our model systems, everybody thinks the neuroendocrine cell is the cell of origin, so they're always introducing their genetic hits in these cells. But maybe small cell is coming from either a tuft cell, and that's why the tumor is tuft-like, or maybe it's actually coming from the basal cell, because the basal cell can give rise to both tuft-like states and neuroendocrine states, and that seems to be what's happening in human tumors. So we took a step back and decided we needed to initiate small cell in these basal cells. And mouse models allow us to do that because you can actually very precisely engineer genetic alterations in specific cells of origin. So these, what we call GEMs, or genetically engineered mouse models, are mice that we've engineered to have the genetic changes found in the human disease. And then we can go and introduce those genetic changes either in the neuroendocrine cell, which is that accepted cell of origin, or a basal cell. And I'm just going to give you an example of, how we do that. So, for example, in the mice, we will introduce loss of RB and P53, because that happens in all of human tumors. And then we can play around with, do we turn on MYC? Do we delete PTEN? There's another gene that's inversely associated, ASCL1. I don't have time to go into that, but suffice to say, we can create all kinds of genetic alterations and then compare what happens in a neuroendocrine cell versus a basal cell. So an example of what the data looks like is that when we initiate tumors in these cells and we stain for ASCL1, NeuroD, and PAL2F3, where brown is positive and blue is negative, you can see that when we start tumors in a neuroendocrine cell, we get ASCL1 and NeuroD, but no PAL2F3. But if we start tumors in a basal cell, we now get ASCL1, NeuroD, and PAL2F3. So we get a different type of tumor depending on what cell of origin we start the disease in. But this cell better resembles what the human data would predict, because the human data says that MYC is high in the PAL2F3 positive tumors, and we only get that here. The other thing we can do is harvest tumors from these animals and do single cell genomics and look at what are the states of every cell in the tumor. And this is just a very colorful example where every dot is a single cell from a tumor. And this is just to show you that we get these different cell states, lots of different cell states, in these mouse tumors that actually highly resemble the states that we find in the human disease, but much more so in the basal cell of origin than the neuroendocrine cell of origin. Okay, so I'm not going to go through any more hardcore data. I'm just going to summarize actually a lot of data to say that when we initiate small cell in a neuroendocrine cell, we get limited states. So when we start tumors in this ASCL1 neuroendocrine cell, we get that ASCL1 and NeuroD state, depending on what genetic alterations we put in. But this really is what we would call limited plasticity, because we're not representing a lot of other states that are found in the human disease. So that model is failing to really recapitulate the breadth of what you see in a human tumor. In contrast, if we start tumors in a basal cell, we now get all of the states highlighted here in these different colors. This ASCL1 state, a NeuroD state, a PAL2F3 state, a hybrid AN state, and even some states that are more basal and less committed to any of these other states. But the most important thing, besides this diversity in the human tumor, or diversity in the mouse tumors that look much more like the human disease, is that when we alter these genes that are found altered in the human disease, we get what would be predicted. And that doesn't really happen so much over here. An example is when we delete P10 in this model, we only get the ASCL1 subtype. But we know that P10 deletion is super common in the Pal2F3 state, and that happens over here when we use a basal cell of origin. So this data has really convinced us that for 40 plus years, we've had the cell of origin wrong. And we think that's really important that we now know where the tumor is arising, what cell type the tumor is arising in, and it's made our model systems better because now the models better recapitulate the human disease. Okay, one other thing I want to say, because we always care about drugs and drug targets and what's going to happen in the clinic. So these findings have also led us to realize that drug targets are highly enriched in specific cell fates, and they're absent in others. So for example, if I show you in single cell format what all the different cell states are that we find in human tumors, color-coded, and then I show you some of our favorite drug targets that you may have heard about, we've heard about today, others you'll continue to hear about, so DLL3, which is that surface molecule that's being used for all kinds of targeting purposes. SEZ6 is another one. TROP2, I think AstraZeneca mentioned. What you can see in this heat map, where dark red is high and white is low, these drug targets aren't expressed in every single cell you might find in human small cell. They're very enriched in certain cell states. We already know this from the literature, but DLL3 is expressed in these states, this ASCL1 high and ASCL1 NeuroD hybrid state. It's a little lower in the NeuroD state and certainly the basal states and other non-neuroendocrine states. Okay, so this is very informative because what it means is there may be cells in the tumor that are in other states that aren't going to express DLL3, and those cells may not be seen by the drug. Moreover, if cells downregulate, shut off DLL3, as they go to these other states, it can help them avoid therapy. And so we want to be able to predict the ways in which cancer cells may seek to avoid the therapy so that we can ultimately prevent it. Now you can appreciate here that SEZ6 has even a different pattern than DLL3. It's really high in this hybrid AN state, and it's less expressed in the ASCL1 specific and basal states. And TROP2 is totally different. It's not really in these neuroendocrine states. It's actually highly expressed in the basal cell state. So this can be very informative for predicting which patients are going to respond, predicting how tumors are going to evolve in the face of therapy. And we really couple this information with very sophisticated technologies called lineage tracing, where we mark cells and follow their ability to shift to these different states, because it's not actually totally random. They do follow some rules as they move from these different states. And so the fact that they follow these rules can help us learn the rules and intercept and prevent their movements to ultimately, hopefully, make for better responses in the clinic. So our lab really thinks that this famous sort of heat map, it's really time to revise this blueprint and to look at these states through the lens of cell fate and the normal cell types that are found in the mouse lung, because these states actually highly resemble, I say mouse lung, also human lung, These states really resemble normal cell types in our bodies. The neuroendocrine cell, neuronal cells, tuft and actually tuft ionocyte-like cells, and this more controversial state we now appreciate is more like an inflammatory basal cell. And this lens can really help us understand the cancer, know its therapeutic vulnerabilities, and ultimately predict how cells are going to shape-shift and evolve. Okay. So if the sciencey part was hard to follow, these are just some take-home messages, hopefully in simple terms. So within small cell, it's not a uniform disease. You have molecular subtypes, and they're defined by expression of these you can call biomarkers, ASCL1, NeuroD, Pal2F3, and YAP. It's true that these are not yet being regularly used in the clinic, but they are being highly explored. There's panels being used currently by MD Anderson for enrollment in clinical trials. They're being, I know, regularly used at Sloan Kettering for research purposes, and I expect to see more and more of this upcoming for enrollment in specific types of trials. We also know these states have very different therapeutic vulnerabilities, and so it really matters what state these cells are in. These states are really mimicking normal lung cell types, and it's important to use that framework to help understand the identity of these states and how to target them. What are they dependent on? What genes are they using? And understanding how they may evolve. And importantly, these states can and do evolve. So we call this cancer cell plasticity, or the ability to change state. And this is obviously a major challenge for treatment because you basically have a moving therapeutic target. And we want to be able to restrict their movement to make them easier to treat. And we want to be able to engage the immune system because the immune system is evolvable, whereas all these therapies we're talking about are very static and don't evolve in your body, which your immune system can. So we really want to get the immune system involved in treatment. And finally, we think we've overturned some very long-standing dogma. We think small cell actually begins in the basal cell. In addition to our work, computational studies are in the process of coming out that use very sophisticated computer algorithms, machine learning, AI types of technology that are very clever and have recently come to the conclusion in human tumors that small cell also arises from a basal cell and not a neuroendocrine cell. And we think this has very important implications for early detection and probably prevention and treatment. So we think that small cell is starting in those stem cells. It may be held in check for a while by the immune system. It may be smoldering for a while, but eventually some event happens that allows those basal cells to escape and become neuroendocrine and be less seen by the immune system and ultimately progress. So now that we have better model systems for understanding this, hopefully we can get closer and closer to how do we detect this earlier, how do we prevent this from progressing and nip it in the bud at an earlier stage. Okay, with that, I want to thank the entire lab. It really takes an army to do this kind of work. And our generous funding sources, you guys may or may not know that the NIH is experiencing a massive funding crisis right now. So your understanding of those issues and your help and support is greatly needed because as I explained in our tour yesterday, this is dramatically hurting research in our country right now. And we need to sort of stop the bleeding as soon as we can. And it really, it takes all of you to help make that happen. So please educate yourself on this topic. Also, we couldn't do our work without tremendous collaborators from all over the country. I've listed some here. We have a phenomenal community in small cell research. Everyone is very generous and sharing of their time and reagents. And I think because of that and because of NIH's effort in making small cell a designated recalcitrant cancer, that has brought together a lot of us under one umbrella, and it's made this research go much faster and more efficient. And that's really thanks to this wonderful government initiative. The number of papers on small cell has exploded in the last 20 years, and this is largely due to the NIH's effort to bring us together and to fund small cell specifically. Now that funding mechanism is currently ended, so that coupled with the NIH challenges, we're going to need ways to stay together and keep the momentum moving forward, and we certainly appreciate your advocacy toward that. And with that, I don't know how much time I have left, but I'm happy to take any questions.
Yeah, I got it. Thank you. Yeah, I thought we were going to hold questions till later, but go ahead. Lindsay,
Oh, yeah, either way.
Oh, Vicky. Oh, I'm sorry. I thought it was you. Jean jackets. I just saw two jean jackets. There you go. Thank you, Vicky.
Hi. So if the basal cells are going to lead to early detection, how do you actually test for that cell? And how do you get that test to the community? I mean, is there a way that we can start saying our GPs at age 50 or 40 that we start screening? And I do want to know what the screening process would be for that basal cell.
Yeah, so it's a great question and something that we have to think about. This is going to be a major challenge, right? Because the surface epithelium of the lung is broad and vast, and we may be looking for a very small number of abnormal cells. And technology right now doesn't exist to sort of scan the entire lung and say, you know, do we have any abnormal pockets of basal cells? But as we begin to understand the process of how does it go from being a normal basal cell to something that is a dangerous, you know, small collection of basal cells, as we understand the molecular mechanisms of how that happens, I can envision having sort of preventative measures where we, you know, sort of intercede and say, hey, we all may have some level of smoldering basal cells from prior toxin exposure or smoke or whatever, and here's something we can do to help prevent that progression from here to here. And then going forward, as those regions get bigger and more detectable by today's technologies, you know, I think we will see improvements in being able to take advantage of imaging or, you know, biomarker-guided imaging to see those things earlier and then intercede. But this is going to take research to get us to that point. Yeah.
I think years.
You know, it could be years. Yeah, I think that this is an important recognition point that's brand new, you know, frankly. Unpublished, but will be coming out soon.
It's exciting, thank you.
Thank you.
Also, can I just ask one more? You're telling about our small cells are many different cells. So, so for treatment, do we need various different kinds of treatment to affect all those cells? Because I mean, I get one treatment at a time, and then we see where that goes, and then we go to another.
You're absolutely right, and I think it's part of why we often see combination therapies are more effective in the clinic than any single therapy, because you do have cells moving to different states, and it can be very complex, and so it's very difficult to have one drug that can, you know, combat all of that movement. And so that is part of what we're trying to do, is design rational combinations that help restrict cancer cells to more uniform, specific states that we can do a better job hitting them. And also, like I said, get the immune system involved. So can we push cells to states that are more likely to be seen by the immune system, so that it's not just our therapy, but the immune system going after the cancer as well. So that's, you know, those are big efforts ongoing right now. Our lab and others are seeking to combine things like the DLL3 and SEZ6 types of drugs that do a really good job hitting neuroendocrine cells, but don't do as good a job hitting the non-neuroendocrine cells, with other therapies, maybe like TROP2 or immunotherapy or other drugs that may do a better job against the non-neuroendocrine states, to get the best combinations. Great question. Okay.
Okay. Does the immune system increase, no. Does the basal to increase the immune system? And also, does it do it after and before treatments?
Okay, some great questions there. I'll kind of speak to what I know. So I didn't have time to show you the data, but we actually know that the basal state is the more inflammatory of all the states, meaning that it expresses what we call MHC, which is something that goes on the surface of the cell that presents antigen and says, it has the capacity to say, I'm foreign, so that the immune system can see it. Whereas the neuroendocrine cells, they don't have a lot of MHC, so they don't have anything on their surface, so they're kind of invisible to the immune system. So this is really important, because we and others have seen that the non-neuroendocrine states express more MHC, like flags on the surface, giving the immune system something to see. So that could be a good thing if the basal cell is presenting something that looks foreign and the immune system can see it and kill it. And we actually think like That's why we don't see more small cell in people, because the basal cell does talk to the immune system, and the immune system often keeps those early stage tumors in check. But every once in a while, something happens in that basal cell that allows it to escape the immune system, go be neuroendocrine, and be kind of invisible to the immune system. Now, on the flip side, there are also basal states that are not seen by the immune system, and some really aficionados in here may know that that's squamous lung cancer. So squamous lung cancer is very basal in identity. It still talks to the immune system. You get lots of immune cells coming, but it's not enough to kill the tumor. So there's also basal states that can be dangerous and bad. And we're trying to really discriminate in our research. What are the basal states that are seen and killed by the immune system? What are the basal states that manage to avoid the immune system? The other place related to your question that comes into play here is that sometimes cancer cells we see during drug resistance. So those of you mentioned non-small cell lung cancer. In the non-small cell lung cancer world, those with adenocarcinoma are treated very effectively these days with some tyrosine kinase inhibitors. So you may know of patients that have EGFR or ALK or ROS-driven adenocarcinoma, and more recently KRAS G12C disease, that get treated really effectively with targeted therapies, and the tumors melt away. But then when they become resistant, they emerge as small cell. So you may hear about this transdifferentiated small cell phenomenon. It's been shown in our research that the cells go through a basal-like state before they convert to small cell. So you can think of as adenocarcinoma backs up to a basal state and then becomes small cell. So that basal state we view as a highly plastic state. It has lots of options, just like the stem cell diagram that I showed you, where it's at the top of the hierarchy. It's got lots of options. So in that sense, it can be a bad state that we want to stop, right? So our work and our challenge is really to figure out how to prevent plasticity, promote states that are seen by the immune system, but without promoting states that just give the tumor lots of options to evolve. Does that make sense?
Hi, Dr. Oliver. Thank you again. Your tour yesterday was wonderful. Your lab, the work that they're doing is fantastic, and we are benefiting from that. So I just wanted to say that. I had a couple of questions, and hopefully they're quick. What is the difference between a biomarker and the protein expression on the cancer cell?
Well, cancer has all kinds of proteins being expressed at any given moment, right? Tens, thousands. What becomes a biomarker, the way you guys use that term, is that it's like actively being used in the clinic to guide something, right? But it always starts at the level of an observation in the lab setting, where we see the expression of something and we do all the experiments to figure out what it means and whether it's valuable. And eventually that observation will move into the clinic and clinical trials and be established, through research as a biomarker. But that takes time, right? So I expect, we're sort of seeing this already, and I expect to see more of it, that things like ASCL1, NeuroD, PAL2F3 may eventually become biomarkers in the clinic. They're certainly being tremendously useful from a clinical research standpoint right now. Everybody that does my kind of work pays attention to them. But until they are being used to actively guide therapy, they won't be like a certified biomarker. But we think that this information, as the research comes along, will eventually translate to that level.
And if this is okay, have you guys noticed in your lab any correlation between like ASCL1 and limited stage versus the PAL2F3 and extensive stage? That's
a great question. There are some controversial studies. I'll point out that Tawfiq Awanikoko's group saw actually that in limited stage disease, there was some high YAP expression in his study. And that could be an indicator that that was actually early stage basal-like cancer. If it was early enough, those cells may have still been very basal-like, which is what our data predicts the cancer starts at, and that those tumors were also very inflammatory, so they had lots of T cells. So almost indicating the immune system was still keeping them a little bit at bay. And then beyond that, all I can say is like there have been multiple studies that PAL2F3 does associate with worse outcomes, but that would be within all extensive stage disease. So for the sake of time,
Great.
shall we?
Dr. Oliver, thank you so much. We are running way behind y'all, and I have a feeling you all need a break